So hello everyone, so here we are again. Today is a holiday, it's a Labor Day, so maybe today we are not making barbecues, but we are working so hard, but we are gonna play hard when all this is over. So let's continue our story on protein and amino acid metabolism. So if you remember, we've been through the digestion of proteins and we saw how amino acids as products of digestion are transported throughout the organisms to reach tissue cells and to be converted to either new proteins, or they can undergo further catabolic processes. So here we are again. So today we are gonna talk about the metabolism of amino acid nitrogen, and we're gonna focus on processes of deamination, transamination, and we're gonna just mention decarboxylation processes because the products of this process are also molecules containing nitrogen, different amines that they are. So let's talk about the overview of the metabolism of amino acids. So it has to be considered as a rather complex bunch of processes, so maybe even more complicated and more complex than it was with the metabolism of carbs and glucose and metabolism of fatty acids and lipids, because you constantly have to have in mind two different pathways and two different fates. So you have to consider the fate of carbon atom, and at the same time, simultaneously, you have to take care of nitrogen atoms from amino acids. If you can see from the slide, most often nitrogen is converted, nitrogen from amino acid released in the form of ammonia or ammonium ion is converted to urea in liver and excreted in urine. On the other side, the most often pathway and fate of carbon atoms from amino acid is their further catabolism and conversion to carbon dioxide and water. So obviously amino acid can be involved in some energy pathways and processes in the body, or those carbon atoms can be converted to some other molecules like glycogen and 3-acylglycerols, which can serve as storage molecules in our body. So we're going to talk today about these pathways of removal of nitrogen from amino acid, and in the upcoming videos we're going to talk about the removal and about the fate and metabolism of carbon skeleton of amino acids, which is really complex. So let's start with the situations, with normal situations. When we discussed about the digestion, we mentioned that the average dietary intake of proteins ranges from 70 to 100 grams per day. And during that normal dietary intake of proteins, most of the amino acids are actually involved in the protein synthesis. If you remember, even up to 75% of amino acids obtained by degradation of proteins are involved and recycled and used for the biosynthesis of new proteins, and only excess amounts of amino acids are converted to some other metabolites. So if we take a look at this figure explaining the situation in a fat state of organism. So here we are. Dietary proteins are digested or hydrolyzed to amino acids, and we saw in previous video the pathways and mechanisms by which amino acids are transported by portal bloodstream to liver. So what happens with those amino acids in the liver? So first, amino acids can be used for the synthesis of proteins in liver, And just remember remember that we mentioned that albumin, blood plasma albumin, is completely synthesized in the liver from dietary amino acids. Also, those amino acids can be used for further conversion to some other essential nitrogen containing compounds like glutathione, like serotonin, like creatine, like heme, and so on, which play a very important role in our organism. They also can be converted to carbon dioxide and involved, but this is not a preferable way, as you know from our previous story on energy metabolism. But excess amounts of amino acids can be converted to either glucose or 3-acetyl coenzyme A and fatty acids and incorporated in the structure of 3-acylglycerols. And that is something that we, I think, mentioned when we discussed the topic on digestion on proteins. So glucose can be converted to glycogen and deposited in the liver, or it can be secreted to blood if the blood glucose levels are low at the time. 3-acylglycerols, which are converted from excess amounts of amino acids, are incorporated in VLDL particles and also secreted to blood. Certain portion of amino acids after the meal are secreted from liver to systemic circulation, and those amino acids enter cells and they are involved in protein synthesis within different tissue cells. But we have a bunch of a different set of events during fasting procedures. So if you take a look at this figure here, so during fasting we know that muscle proteins undergo cleavage processes, they undergo degradation and hydrolysis to produce amino acids. Some of those amino acids, if you follow me through this figure, are partially oxidized in oxidative processes as intermediates of 3-carboxylic acid cycle to obtain certain amount of energy. Certain portion of amino acids is converted to alanine, which is secreted from muscles to blood, and some of the amino acids are secreted to blood without undergoing any changes. So we have the conversion of certain portion of amino acids to glutamine and alanine before released into the blood. What is the fate of glutamine synthesized? It can be further oxidized in certain tissues like lymphocytes, gut, and kidney. So if you see here, in kidney it undergoes deamination and it can be converted to alanine in the final stage, and glutamine can be also converted to alanine in the gut. Alanine and other amino acids, they reach liver where they undergo several changes. So amino group is removed to form urea, which is excreted in urine, and the rest of the carbon skeleton can be converted to either glucose molecule or ketone bodies. Glucose can be secreted if the blood glucose levels are low, so glucose is going to be used by brain and red blood cells, and ketone bodies can be used by muscles, kidneys, and to final extent brain again. So this is the overall pathway of our metabolism of amino acids, and we will go step by step. But this time we're going to focus on the removal of nitrogen from amino acids. And there are some enzymes which are important, and they are involved in those interconversions of amino acids and removal of nitrogen. Those are certain different dehydratases, transaminases, glutamate dehydrogenases, glutaminases, and deaminases. Generally speaking, two major pathways for nitrogen removal or amino group removal from amino acids are known, and we're gonna go into details explaining their mechanisms and purpose, and those are transamination and deamination process. So let's start with the first one. Let's start with transamination. Transamination is considered to be the major process for removing nitrogen from amino acids. And actually, it describes the transfer of amino group from one amino acid to another alpha keto acid, so that amino acid becomes alpha keto acid, and alpha keto acid, upon acception of amino group, becomes amino acid, which is presented here in the figure. Enzymes which catalyze these transamination processes are marked as transaminases or aminotransferases. The coenzyme which is required for these processes is pyridoxal phosphate, which is produced upon the modifications of vitamin B6 or pyridoxine. Actually, in theory, all amino acids may undergo transamination process, but in reality, in the field, in a cell, alpha keto acid, which is always the acceptor of amino group, is alpha ketoglutarate. Upon the acception of amino group, alpha ketoglutarate is converted to L-glutamate. And here you can see one example in a figure. For example, aspartate gives away its amino group to alpha ketoglutarate. Thus, alpha ketoglutarate becomes L-glutamate. This reaction and all transaminations are readily reversible reactions. All amino acids I have already mentioned may undergo transamination, but two amino acids cannot undergo transamination under any kind of conditions, and those are lysine and threonine. That is the reason why those two amino acids are considered to be ultimately essential. What about the mechanism of transamination? So it's explained in this figure. So what is going on? Amino acid as a substrate binds to the enzyme, forming enzyme-substrate complex. Then the chemical reaction occurs between the amino group of amino acid and aldehyde group of pyridoxal phosphate coenzyme. The result of the reaction is the release of one molecule of water and the formation of a double bond between the nitrogen from the amino group and carbon atom from aldehyde group. In the next step of reaction, we have the shift of the double bond, so it's now moved to the place between the alpha carbon atom of amino acid and nitrogen atom from amino group of amino acid, and catamine or aldimine is formed as an intermediate product. In the third step of this process, the molecule released in the step number 1 is now the molecule of water released in the step number 1 is now incorporated in the molecule, thus producing alpha keto acid and pyridoxamine phosphate. So if you see from this figure, you can notice that this coenzyme is changed. So enzyme itself is changed during the reaction, which is actually the opposite from what we have thought so far, knowing the general principles of enzymology. But be careful here that this is not a permanent state. It's just temporarily change, because the reaction is absolutely and completely reversible. So pyridoxamine phosphate remains active, but for the reversible reaction. So it can give away its amino group to a specific alpha keto acid, which is alpha ketoglutarate, and the sequence of reversible reactions occur, so you get an amino acid the other amino acid, in this case alpha L-glutamate, formed. So we mentioned that all amino acid in general may undergo transamination, and they really are undergoing transamination, apart from those two, lysine and threonine, that we have mentioned as exceptions. But out of all those amino acids, 3 amino acids and 3 corresponding keto acids undergo very intensive transamination processes. And those amino acids and their keto acids are glutamate and its keto acid alpha-ketoglutarate, aspartate and its keto acid oxaloacetate, and alanine and its keto acid pyruvate. So these interconversions are catalyzed among those 3 amino acids and subsequent keto acids are catalyzed by 2 types of transaminases. The first one, and those 2 are so famous, and we have discussed a lot about these enzymes throughout both theoretical and practical classes. So alanine aminotransferase, ALT, and we have the old name glutamate pyruvate aminotransferase. Sometimes it's still used. Aspartate aminotransferase is the other one, or called glutamate oxaloacetate aminotransferase, GOT. You will see the reactions in the upcoming slide because of the importance of those 2 enzymes in medicine and diagnostics and laboratory. So, but it is important to emphasize, and you can see it from those old names, that there is no direct transfer from, for example, alanine to oxaloacetate or from aspartate to pyruvate, but all of the available transfers are from alanine to alpha-ketoglutarate and from aspartate to alpha-ketoglutarate. So the mandatory mediator is always pair alpha-ketoglutarate L-glutamate. So here are those reactions catalyzed by AST and ALT. So L-aspartate gives away amino group to alpha-ketoglutarate to form oxaloacetate and L-glutamate. ALT catalyzed reaction catalyzes the removal of amino group from L-alanine to alpha-ketoglutarate to form pyruvate and L-glutamate. So we have mentioned that all other amino acids also undergo transamination processes, but the difference between these 2 and the rest of the amino acids is in the font of available alpha-keto acids and the way or in the possibility of their synthesis. Why is that so? Because pool or reservoir of all other alpha-keto acids is limited by the pool of the corresponding amino acid. So you can transaminate amino acid to its alpha-keto acid, and then you can convert that alpha-keto acid to amino acid if that is necessary at the moment for the cell. But the overall amount is not increased. It remains constant. It is especially important and valid for essential amino acids because our body cannot synthesize those amino acids, and they can be obtained only through diet from dietary sources. So it's not likely that those essential amino acids are gonna undergo frequent transamination, if they are gonna undergo transamination at all. So, but on the other side, pool of the mentioned 3 alpha-keto acids and 3 amino acids is far more larger, far more bigger, because those alpha-keto acids that we have mentioned can be obtained from other metabolic pathways of some other biomolecules, like from the metabolism of carbs or from metabolism of lipids indirectly. So we have we all know about alpha-ketoglutarate and oxaloacetate as intermediates of Krebs cycle. So there are significant amounts of those alpha-keto acids available from other sources, from carbs entering Krebs, from fats entering Krebs, or pyruvate, which is an intermediate or the final product of glycolysis. So glucose can be converted to pyruvate, and pyruvate can be used for transamination by ALT and converted to alanine to a significant amount. Processes of transamination are of great importance because they actually enable replenishment of deficit amino acids at the expense of those amino acids which are found in sufficient amounts in the body. So if our cells are lacking certain amino acids, they can replenish its quantity because they can take away amino groups from some other amino acids which are in excess amounts and to form those lacking amino acids. So deamination process is the second way to remove amino group from the amino acid, and this is an oxidative process. Upon deamination, corresponding alpha-keto acid and ammonia or ammonium ions are formed. Most often, again, in theory, theoretically, each and every amino acid may undergo deamination process, but in reality, most often that is L-glutamate. Maybe it's the only amino acid undergoing deamination process in organism. So L-glutamate is deaminated to form alpha-ketoglutarate and ammonium ion or ammonia, and the reaction is presented here in a figure. Those deamination processes take place most intensively in the liver and in the kidneys. And there are two groups of dehydrogenases catalyzing these reactions. In aerobic conditions, in the presence of oxygen, those are aerobic dehydrogenases requiring flavin mononucleotide as a coenzyme, and in anaerobic deamination processes, coenzyme NAD or NADP plus equally are required. So let's go through the mechanisms of those. So in aerobic conditions, amino acids bind to an enzyme to form enzyme substrate complex. And then reducing equivalents, those hydrogens from alpha carbon atom and amino group are then transferred to flavin mononucleotide coenzyme, thus forming double bond and amino acid molecule. The reaction is catalyzed by aerobic dehydrogenase. The second reaction is spontaneous non-enzymatic hydrolysis of amino acid to alpha-keto acid, and ammonia is released from the system. Reduced FMN is regenerated in the presence of molecular oxygen spontaneously to form FMN again and hydrogen peroxide. And we know very well that the subsequent fate of hydrogen peroxide is to be reduced by either catalase or peroxidase to water and nascent oxygen. In anaerobic conditions, I have provided you with the example on L-glutamate here. So L-glutamate undergoes anaerobic dehydrogenation, and again reducing equivalents are transferred to NADH coenzyme to form NADH coenzyme, and we get alpha-aminoglutaric acid. NADH coenzyme is regenerated in the way that it releases its reducing equivalents to electron transport system of mitochondria, and alpha-aminoglutaric acid, like previously, undergoes non-enzymatic hydrolysis to form alpha-ketoglutarate acid and ammonia. So these processes occur in mitochondria, and like transaminations, they are readily and absolutely reversible reactions. Now that we know all about transamination and deamination processes, we can make a connection between those two processes because actually they act together, they act simultaneously. So we heard previously that during transamination processes, amino acids from our amino acid pool can give away their amino groups, and glutamine is alpha-ketoglutarate is the molecule which can collect amino groups from other amino acids to form glutamate. And glutamate can now undergo direct deamination by glutamate dehydrogenase to release ammonia. So if you have that in mind, the overall picture in mind, you can conclude that actually transamination itself is a type of indirect deamination process. So as a result or as a product of deamination processes, toxic ammonia is formed. It's released from deamination reactions, and ammonia as a toxic molecule, especially for nervous system and brain, is readily transported. We will see the patterns later on in the next video. It's transported to the liver to undergo detox processes to form urea, which is going to be safely excreted in urine. So glutamate dehydrogenase, the major enzyme in deamination process, is actually considered to be one of 3 available enzymes that can actually fix or bind toxic ammonia or release it when necessary. Other 2 enzymes are glutamine synthetase and carbamoyl phosphate synthetase, and we're gonna mention those enzymes and see their catalytic function later on in the upcoming videos. So, apart from this deamination of glutamate to alpha-ketoglutarate and ammonia, in our body there are some other reactions going on which also yields the formation of ammonia or ammonium ions. So there are some other sources. Let's go through them and let's prepare ourselves for the topic for the next video. So there is a certain balance and equilibrium between ammonia and ammonium ion, and a lot of authors are actually overlapping and mixing those 2 terms, whether to say ammonium or ammonia or whether to say ammonium ion. But this equilibrium sets the pK constant of 9.3. So in under physiological conditions in the body, the equilibrium is shifted towards ammonium ion. And there is an important difference between those 2 forms. Ammonia can diffuse freely through membranes, but ammonium ion cannot. And this is important, for example, in processes of acid-base maintenance, equilibrium maintenance, because ammonia can diffuse from tubulocytes to urine, and in urine ammonia absorbs secreted hydrogen ions to form ammonium ions, and ammonium ions cannot be again reabsorbed and pass through the membrane back to the cells. And there are some other examples, of course, as well. But if we take a look at this figure here, it explains beautifully all the sources of ammonia and ammonium ion in our body. So let's list those 6. The first one is the one that we have explained. So it's glutamate undergoing a deamination process catalyzed by glutamate dehydrogenase in the presence of NAD coenzyme to form alpha-ketoglutarate and ammonia. The second source of ammonia is direct deamination of histidine to form urocanate and ammonia or ammonium ion. And we will see those details in the upcoming videos. Here is just the list to see with what we are actually dealing with. The third source of ammonia is metabolism of 2 amino acids containing hydroxyl group in the structure, and those are serine and threonine. They undergo both deamination process and dehydration process, and in both cases pyridoxal phosphate is a required coenzyme for the transformation, and the enzyme is serine dehydrogenase. So in this transformation, serine is converted to pyruvate and ammonia, and threonine is converted to alpha-ketobutyrate and ammonia. Again, details in the upcoming videos. What else do we have here? Number 4, neutral amino acids, glutamine and asparagine. You know that they have amido group in the side chain, so they can undergo so-called deamidation process with enzymes glutaminase or asparaginase to form glutamate or aspartate, and in both cases ammonia. And finally, number 5, not finally, but number 5, muscle and brain. I'm not going to go into details because this is something for some future stories, but you can just memorize here that during purine nucleotide cycle, in both brain and muscle, aspartate is used up to form fumarate and some necessary parts of the purine ring, and again ammonia is released from the system. And finally the gut. So in our intestine we have bacteria active, intestinal bacteria active, and those bacteria can convert amino acids to ammonia and various products. They can turn urea to ammonia and carbon dioxide, and ammonia is then reabsorbed from intestine and by enterohepatic circulation transported to liver to form urea again. And finally, to conclude this lecture with just the short notice story on decarboxylation of amino acid, which actually means the removal of carboxyl group from the amino acid. This is a process which occurs to a really lesser quantitative extent. And as a result of decarboxylations, amines and carbon dioxide are produced. And I have provided you with one example. So histidine undergoes decarboxylation by histidine decarboxylase to form histidine, which is a very well-known amine mediator on inflammatory response of our body. So anyway, that was it for, that was all about the metabolism of nitrogen from amino acid and formation of different nitrogen compounds. Now we have to see how those ammonia and ammonium ions are actually converted to urea and detoxified. This is going to be the topic for our next video. So until then, please enjoy this day, work hard despite it's a Labor Day, and see you next time. Bye bye.